order. It's our pleasure this month to have Angie Laird from Florida International University. Angie is amongst the many physicists by original training of us out there. Do physics first, ask questions later. It's kind of some of our mantras, perhaps. So she got her BS in physics from Florida State University, PhD in physics and medical physics from the University of Wisconsin at Madison. She is a faculty at the Research Imaging Institute at the University of Texas Health Sciences Center at San Antonio, where many of us ran into her a bunch. And ever since then, she's sort of been part of the greater ReproNim team and was PI of the ABCD ReproNim course, and all around great collaborator in many things ReproNim. So today she's currently a professor in the Department of Physics, director of the FIU Center for Imaging Science, and will be speaking to us today on large open data sets for neuroimaging research. Thank you, Angie, for joining us.
Thank you, Dave, as always, and thanks for the invitation. Glad to be here today. So I am going to talk to you a little bit about large open data sets for neuroimaging research with an emphasis on considerations for reproducible and responsible data use. I have geared this talk to be fairly at an introductory level, so I know a lot of the folks that will be watching either now or later come from more advanced backgrounds, but I think there'll be some ideas to stimulate interest from all of us regardless of background. I've organized this talk based on a review paper that I wrote for Neuroimage back in, actually I wrote it during lockdown in the summer of lockdown in 2020. And it was just sort of the way ideas were evolving my brain of thinking about all of the large open data sets that are available to us. So I started writing the paper, and to be honest, the paper that I ended up writing wasn't exactly what the editors asked for. They wanted a review of the data sets themselves. And frankly, I found that a little bit boring. I did start with a table of I tried to do what they asked me to do. So I did start with a table as shown here of the data sets that were sort of catching my eye and how I was thinking about going through them in terms of groups from large, healthy young adult data sets, lifespan data sets, dense sampling, and data sets featuring many tasks and experimental measures. But as you know, the availability of these data sets out there is pretty wide open and vast for us, so this is not a comprehensive listing. But as I started writing the paper, I kind of really hit my stride and started talking about the things that matter a lot to me right now, which is how we can use these large neuroimaging projects to address very urgent issues related to diversity, equity, and inclusion in our research community, and what are the responsibilities that we bear when engaging ethically with these data sets. So before we get sort of properly into this, I want to give everybody fair warning that I'm a big fan of the Marvel Cinematic Universe, so I have various little animations and whatnot scattered throughout the slides, so just so you know that'll be coming. So an increasing body of evidence points to some reproducibility issues in biomedical or life sciences, and psychology and neuroimaging in particular are currently experiencing a replication crisis. So we, as in our collective community, have published a number of different papers about this. And I've listed, some of my favorites here. A good bit of this discussion has also found its way into mainstream media outlets, which definitely complicates the ongoing discussion. But as one of my favorite all-time tweets really perfectly captures, I don't want to talk about the dead salmon anymore. We know reproducibility is an issue. We know there are a number of different issues in the neuroimaging community. What I do want to do is focus on the future and how we can address this crisis moving forward. So I actually like to reframe this as a reproducibility opportunity. Sure, we know that some of the things that we've done in the past haven't held up very well, but that's okay. Let's focus on the future and what we can do moving forward. So over the past several years now, our community has collectively been working on strategies for improving the reliability, the reproducibility, and the replicability of functional neuroimaging studies. So let's talk just a little bit about the resources that are available to help us with that. So helping us focus on these 3 R's, we have the infamous Spider-Man pointing meme, which has actually been recently replicated itself multiple times. So There's a number of different resources available to neuroimagers, which I like to think of as divided into 3 different categories, the first of which is guidelines and recommendations. First, I want to highlight a 2016 report developed by OHBM, the Organization for Human Brain Mapping, Best Practices on Data Analysis and Sharing. This is the COBITAS report that gives researchers clear and detailed guidelines and standards for promoting reproducible research. In addition to this being a very comprehensive document that covers the lifespan of a data set, the COBITAS report also includes a really great table at the end that addresses reporting of experimental design, data acquisition, pre-processing, and statistical modeling. It is often really easy to forget some of the details of this when you're rushing to get a manuscript drafted and out the door to your co-authors and submit it to a journal. So what I'd like to do is keep this document open to that table at the end when I'm drafting my method sections to help remind me, to prompt me of all of the pieces of information that I need to include in my manuscript. So next, I want to encourage everyone to read a pair of papers that gives a practical and accessible overview of open science research practices for making research more reproducible and transparent. And then this pair of papers gives with regard to large open data sets, a lot of hands-on details and suggestions. The first paper here is broadly about working with large-scale open data sets, and the second is a preprint that's been recently published by Developmental Cognitive Neuroscience that focuses on issues specifically related to working with ABCD study data. This one's really excellent. So finally, as datasets increase in size, researchers are faced with concerns that their institution's local computational infrastructure may not be sufficiently robust to handle data from thousands of participants. And this is a huge shift from maybe the old or even not so old days of running analyses on our desktops and laptops. And so I'm highlighting here a pair of papers that discusses resources for science in the cloud, which encourages users to use containers, cloud computing, and cloud data services to conduct large-scale data analyses of neuroimaging data. And the second paper here focuses on using Amazon Web Services, or AWS, to run neuroimaging workflows and provides helpful instructions for benchmarking time and estimating costs, which we'll talk a little bit more in an upcoming slide. And then finally, I want to highlight a unique but really super helpful resource. The Horian et al. Paper here is actually the one at the bottom left is a traditional journal article, but they've also published a companion resource as a STAR protocol. And this is a recipe style protocol that doesn't waste any time with large chunks of descriptive text, but instead really gets into detailed step-by-step for processes of what you need to do and consider when you're working with large open data sets. It's really a practical resource, and I urge you to check it out. The second category of tools and resources focuses on standard software and data management. We could do a whole seminar just on this, and I think there are ReproNim seminars just on this. So my goal here is not to really go into any great detail, but merely highlight what I think are the go-to resources for us all. So I've included neuroimaging data standardization, which begins with clear specifications for file formats and also includes data organization and data specification. Our community has wonderfully seen the emergence of turnkey data processing and analysis software tools like DataLad, which is available for data management and versioning. And additional tools that me and many others have become part of our daily lives include those for text and code, data visualization, and project management. And then finally, the third category of resources relates to training and education. For me, one of the most exciting and rewarding aspects of the open science movement is the diverse range of training opportunities that have sprung up in various places where researchers come together to learn data science skills and work on collaborative projects. So these include the Brain Hack Organization, Neuro Academy, NeuroMatch, the OHBM Open Science Special Interest Group, the OSIG, and of course, as you all know, ReproNim. I'm really impressed by the community building vibe of these groups, and I encourage everyone, if you're unfamiliar with any of these, go ahead and check them out. And then I also have a quick little note here for a collaborative course that sprung from the ReproNim Foundation. That was our ABCD ReproNim course that we offered the last few years to learn about the ABCD data set. This course has ended, but the course materials are all online and remain accessible for others. Okay, so I want to segue a little bit now and talk about what I think are some of the compelling issues in our field today. And for that, I really want to jump into the issues related to the analytic multiverse. I think that this is something that we're all sort of starting to grapple with. And so I want to introduce and kind of lead up to what I think are the big issues for us. So let's start our journey into the analytic multiverse. And the first step into this journey begins with Josh Karp's paper. This came out all the way back in 2012. The paper was called On the Plurality of Methodological Worlds. And this paper revealed to us that the choices that we are making when constructing our analytic pipelines might actually be influencing some of the outcomes of our results. So in this paper, data from a single event-related fMRI experiment was analyzed using multiple different analysis methods. 10 different steps were identified, each with 2 to 4 different strategies that yielded nearly 7,000 unique analytic pipelines. Activation maps were generated and then corrected with 5 different thresholding approaches, yielding nearly 35,000 maps. The results here showed that, in general, estimates of overall peak activation strength and statistical significance were highly variable across pipelines. So then fast forward into 2020, when the NARPS paper was published. NARPS stands for Neuroimaging Analysis, Replication, and Prediction Study. And in this study, 70 independent teams analyzed the same fMRI data set to test the same 9 hypotheses. Notably, no 2 teams chose identical workflows. So out of 70 groups, everybody had different ways of doing things, demonstrating that researchers have many degrees of analytic freedom. And this flexibility resulted in sizable variation in the results of the hypothesis tests. On average, across the 9 hypotheses, 20% of teams reported a result that differed from the majority of the teams. And this variation was observed even for teams whose statistical maps were highly correlated at intermediate stages of the pipeline. Overall, analytic choices have a major effect on reported results. Across teams, variation in results was related to several aspects of the analysis methodology, which I'll review in the next slide. So the NARPS paper showed us that even if we take the same data set and want to test the same hypotheses, then different groups are likely to end up with different answers. It is hard, if not utterly impossible, to estimate reproducibility using a single analysis pipeline. That is, the array of analytic approaches that are available to us is so vast and flexible that it's our own analytic multiverse, as illustrated here by our Loki slides. So from the CARP and the NARP studies, we know that key stages are more likely to influence results, and those are spatial smoothing, multiple corrections, software packaging, and denoising. But I also want to highlight that we have a lot of choices to use when we're parcellating brains into regions or networks using a structural functional atlas. And for this point, I want to highlight a cool paper. Well, this is by Ruby Kong. She presented it at OHBM last summer. I think the paper is in works. But this looks at spatial correspondence across 16 group level functional atlases. I believe that collectively this work is really going to help us make some progress in resolving some of the unknown variability associated with there being so many different atlases with computing nomenclature that are in use across studies. But anytime you talk about analytic flexibility with another researcher, the response is nearly always, okay, it's great that we know about this, but what are we supposed to do about it as individual users? Or, as we see in this little graphic here, what is reviewer 2 going to say about what I'm doing from the perspective of analytic flexibility in my paper that I've just submitted? And to be clear, I don't think that there are any solid answers yet to this question, but I do know that the first step is to talk about this broadly as a community and to be fully transparent in your papers about your pipelines and the choices. This point was articulated very well in a quote from the CART paper, again, back from 2012. To mitigate the effects of this flexibility on the prevalence of false positive results, investigators should either determine analysis pipelines a priori or identify optimal pipelines using data-driven metrics. If researchers use multiple pipelines to analyze a single experiment, the results of all pipelines should be reported, including those that yielded unfavorable results. There's a lot to unpack there. I really like this quote. I think it really sets out some very helpful guidelines for all of us. And then there was a tweet on Twitter that caught my eye that makes my next point. What I really like most about the brain mapping community is the interaction between the applications folks and the methodologists. The community itself is just amazing. We have a lot of amazing folks in our midst that spend their entire careers in informatics and software development, working tirelessly on tools that are critical for advancing brain science. And what's next on their agenda is helping researchers make multiversal pipelines accessible for all of us. So my advice is to recognize that our prior work may not be as robust and as reliable as we thought at the time from the lens of analytic flexibility. But what matters now is that you know this and that you do your best in producing good, solid, transparent research that leverages the very best resources for open and reproducible science that our field has to offer. And that we all sort of hang tight while some of these newer tools for multiversal analyses are developed and introduced to our community. So next I want to transition a little bit and talk more about our neuroimaging research community. As you all know, MRI research is expensive, and historically, your institution's ability to purchase and maintain access to an MRI scanner determined whether or not you could participate in analyzing fMRI data to study brain function and organization. But the advent of large open data sets has ushered in a new era for us all. These data sets now represent centralized resources that may be accessible by a wider pool of researchers, thus circumventing local resource limitations and broadening research opportunities. So, this democratization of fMRI research has the potential to increase capacity for equity among researchers where everyone has equal access to data, as well as the computational infrastructure to analyze it. But hang on, what about those computational resources? Remember how I mentioned cloud-based computing a few slides ago? Some researchers have pointed out that advances in cloud computing may further broaden accessibility among researchers who lack resources such as high-performance clusters at their institution. However, another viewpoint is that while cloud computing costs associated with the pay-as-you-go model may be considered reasonable and affordable to some research labs, they still remain prohibitively expensive for others, particularly when analyzing large data sets that require many hours of analyses. And it's important to note that the discussion of cloud computing thus far has really centered around services provided by private US-based corporations, including AWS and Microsoft Azure cloud services. So moving forward, enhanced resource access through the equitable availability of computing resources may be more fully realized through federated computing grids. An example of this is Compute Canada, or decentralized analysis platforms such as CoinStack, or nonprofit organizations such as the 2I2C managed Jupyter Hubs. But the issues of diversity and inclusivity are relevant not only among scientists who are conducting the research studies, but we should also pay important attention to the people who are asked to participate in those studies. And I think that this is really a critical issue for us. It's been 10 years now since Henrik et al. Pointed out that research studies are overly reliant on weird samples. And if you're not familiar with that acronym, it stands for Western, Educated, Industrialized, Rich, and Democratic. Although weird samples are not representative of global populations, results from weird studies are frequently assumed to generalize to all humans. And then you have neuroimaging specifically, which is a very technically challenging field. It requires significant preparatory work in developing hardware protocols and imaging environments for data acquisition, and this work has historically been led by physicists and engineers. And as a result, we have a predominance in neuroimaging of convenience samples in the literature, which are comprised of American or European university students who are frequently affluent, white, cisgender, right-handed, and healthy. Such samples really reflect more on the challenge that we all face in meeting the essential methodology of imaging research, and less on efforts that were spent to ensure recruitment of an appropriately diverse and representative sample. And so the result is, when you look at collectively what we've done so far in the literature, it's no wonder that the more recent studies that look at Western versus non-Western cultures or sociodemographic status reveal huge issues with reproducibility and generalizability in our field. So I'll really be honest here. I read a lot of papers that emphasize the rigor of their recruitment strategies, and then I go look up the demographics of their sample, and it's a little bit of the channeling of the Agatha meme. I just think that we can all do a little bit better, put a little more effort into this. And so what I would like for us to consider is that we are collectively generating a large body of literature that is derived from relatively not very diverse group of participants. And so, in the interest specifically of reproducibility and generalizability, I think we need to rethink our participant recruitment strategies and also start to take into consideration the study of brain function as a global endeavor. So when many of us think about large open data sets, we immediately think about the big three: the HCP, the ABCD, and the UK Biobank. A lot of us use these data sets. But while I was writing the review paper that I mentioned at the start of this talk, I spent a good amount of time learning more about the Cuban Human Brain Mapping Project, India's Consortium on Vulnerability to Externalizing Disorders and Addictions, or CVADA, Brazil's high-risk cohort study for the development of childhood psychiatric disorders, the Japanese ADNI project, the Australian Imaging Biomarker and Lifestyle Flagship Study of Aging, and from China, we have a number of multiple exciting data sets. And so finally, I want to end this slide by noting that the advent of population-level neuroscience really provides an opportunity for neuroimages, wherever they are, to collaborate with social epidemiologists to address the lack of representative samples and strive for more inclusive enrollment strategies among more diverse and underserved participant groups. So for example, in the ABCD study, close attention was paid throughout enrollment to the emerging demography of the ABCD cohort to strive for broad diversity across multiple dimensions, including race, ethnicity, family income, and parental education. When the investigators observed that the sample included an overrepresentation of high-income families who had completed more years of education, additional resources and support, yes, more funding, was provided to sites during the last 6 months of enrollment to prioritize recruitment of low-income families with fewer years of education. So what I want to say here the most is that I think that the inclusion of racially, ethnically, geographically, and socioeconomically diverse samples is likely going to help all of us and result in the more comprehensive understanding of brain behavior mechanisms, particularly through the lens of cross-cultural global mental health. So next, I want to shift a little bit more and talk about the responsibilities that we carry as researchers who engage in large open data sets. Pretty much every movie and show in the MCU plays a little bit with a theme about the balance of power and responsibility. So here we have Captain Marvel illustrating the point here. The traditional training that we all receive in responsible conduct and research emphasizes critically important topics, but they relate primarily to activities prior to and during data collection, like protection of human subjects, animal welfare, and laboratory safety. But responsible conduct research also extends to data management, sharing and ownership, scientific rigor and reproducibility, and responsible authorship and publication. And an increased sharing of large open data sets must be accompanied by heightened attention on participant privacy, including individuals from more vulnerable populations, such as patients with clinical disorders and/or historically underrepresented groups. And then beyond concerns about participant privacy, responsible data analyses require careful planning, becoming familiar with data acquisition protocols, and understanding the limitations of the acquired data. Generally, when you're planning to collect study data, Scientists can choose the research instrument that best addresses the research question. However, when you are working with existing data, the study design cannot be manipulated. So consequently, the scientific process is actually reversed, and the scientific question must be designed based on the appropriateness of the available instrument or measure. And the truth is that when you don't have to collect data, your projects just move faster. You come up with ideas, you look at the data, and you start analyzing. But it is really up to all of us to take a moment to pause before engaging with the data, think carefully to ensure that we're asking research questions that are suitable for that given data set. Now, prior to any data analysis or interpretation, researchers must engage responsibly and fully consider the psychological, social, economic, or any other potentially harmful impacts their research could have on individuals, communities, and society. Specifically, this means we need to thoughtfully consider the responsible use of variables related to race, ethnicity, gender, and sex when conducting analyses of neuroimaging data. Neurocomparisons across participants who are grouped by race and/or ethnicity can unintentionally provide support for a biological deficits framework, which argues that lower rates of achievement and/or poorer outcomes among minoritized groups are the result of so-called biological determinism. So to discourage this, it is imperative that we as data analysts recognize that ethical conduct in research includes ensuring that analyses prevent further stigmatization, marginalization, and injustice towards individuals because of racial, ethnic, or gender status. Now, let's dig into this just a little bit. Factors that may contribute to stigma in research include a lack of cultural sensitivity or competence about the study design or insufficient understanding of the limitations of the dataset, improper scope, such as not including relevant variables or inadequate context, failing to contextualize, leading to overgeneralization of findings, overstating the significance of findings by making inaccurate assumptions, or insufficiently acknowledging the limitations of the study. There are steps that you can take to prevent stigmatization of individuals or groups as a result of your research, and these include, but are not limited to, consider study inclusion criteria and do not exclude certain groups out of convenience or habit. Respect of people who, from the basis of your research, understand and appropriately and consider how your research might be misinterpreted, not because you intended it to be so, but because others did, to the detriment of those groups. Consider the full range of factors, both positive and negative, that may be related to research results, and situate your research in the appropriate context. Collectively, these practices are critical not only for health disparity research, but for all science. So the information on this slide was adapted from the All of Us Responsible Cognitive Research. For those of you who don't know, All of Us is NIH's Precision Medicine Initiative. And given the large volume of personal and sensitive data that they are collecting, researchers must complete training prior to accessing the data regarding the challenges of conducting research among diverse participants and all the ways that this research might harm those historically marginalized groups. So I encourage everybody here to check out their website and download the training materials. And then I want to note 2 additional readings. I mentioned them on a previous slide, and I'm bringing them back up again because I think they are relevant in this context. The ABCD study, because of its large and demographically diverse sample, it's really leading the way in terms of highlighting important considerations for preventing stigmatizing research. And these are 2 papers that describe these considerations and provide recommendations for how to responsibly engage with the data. So the last point I want to make is with respect to thinking about the future of neuroimaging research and keeping in mind that this is a global endeavor. Historically, neuroimaging studies have gathered basic demographic data that relates to race, ethnicity, and sex gender. And even a cursory literature search will reveal that indeed there are studies that have looked at differences in brain structure and function along these demographic boundaries. And it makes sense, as scientists, we're naturally curious to learn more about this. But what I want to emphasize today is what we're really interested in are the social determinants of health. The World Health Organization defines the social determinants of health to be the non-medical factors that influence health outcomes, including the conditions in which people are born, grow, work, live, and age, and the wider set of forces and systems shaping the conditions of daily life that have an important influence on health inequities. So examples of the social determinants of physical and mental health include income, education, employment, food insecurity, housing, early child development, and social support. So, while neuroimagers have acquired basic demographic data in our past studies, this is an area we all need to do better in. Statistical models that appropriately contextualize findings through the considerations of the social determinants of health, they're not possible unless studies are designed to acquire relevant measures of interest. Most data sets include some variables related to education, employment, and income. This is something that's fairly standard that we see in terms of our data acquisition protocols. But even with these, a recent, well, not really recent, but a pre-registered protocol on OSF, the Open Science Framework, does a really great job of pulling back the curtain and exposing some of the concerns here. This pre-registration reviews the lack of common measurement standards, all the challenges with how we're collecting data on and aggregating these variables and constructing scores that relate to socioeconomic status. And this can lead to a lot of ambiguity and confusion in the field because of the lack of standardization. Other studies, like the ABCD study, are collecting additional measures beyond education, employment, and income. These measures of social determinants of health, the ABCD is collecting a part of its culture and environment protocol. These include neighborhood safety and crime, family and school environment, and ethnic and racial identity. And while ABCD is also measuring parent and youth acculturation, as well as youth perceived discrimination, there are real limitations to these data, and more work is needed to capture a more full and complete understanding of the role that things like cultural stress may play. As a researcher in Miami, my community is largely made up of folks who have immigrated here from the Caribbean, Central, and South America. So these are issues that I think about on a daily basis about our research populations. And then as the scale and scope of population level neuroscience is increased, scientists responsible for study design are really encouraged to consider the inclusion of more comprehensive measures that assess the social determinants of health as a core strategy for understanding how adverse and protective factors impact individuals because of biological, cultural, and environmental factors at individual, community, and societal levels. These are complex phenomena that we're talking about, and it really requires thoughtful data collection. And so for anyone who is thinking about how you can add some of these measures to your own studies and may be feeling a little bit overwhelmed, my final point is simply to say that intentional consideration of the social determinants of health can be accomplished through collaboration involving researchers with expertise in studying cross-cultural psychology and/or health disparities with expertise in these areas. These folks have spent their entire careers thinking about how to use these measures, testing them, validating them, really ready to work to make sure that we have the data that we need to more fully contextualize neuroimaging results. So I want to thank everybody for listening today. It's a little bit of a different talk, hardly any data, and certainly more Marvel references than you're used to. But I do think it's important to sometimes take a step back from analyzing data and really think about some of neuroimaging's big issues. And so as a summary, I started this talk with an overview of the history and evolution of large open data sets. I think it's important to acknowledge how far we've come in the past two decades. I acknowledge the reproducibility crisis and give you lots of pointers to available resources for making your own research more reproducible and transparent, including guidelines and recommendations, standard software and data management tools, and training and education programs. And then we talked a little bit more about equity among researchers, making study recruitment more inclusive, yielding more diverse participant samples, and the responsibilities that we carry when engaging with large open data sets, and specific recommendations for preventing stigmatizing research when dealing with variables related to race, ethnicity, gender, and sex. All of which led me thinking about the future for us all and a call to action for us as a research community to be more thoughtful and intentional about designing studies that include measures that relate to the social determinants of health so that we can more fully contextualize neuroimaging findings that might otherwise incompletely or inaccurately be attributed to biological differences in race, ethnicity, or gender. So we have so much data that's available to us. It's a very powerful and exciting state to find ourselves in. I will quote from a decades-old quote from Spider-Man. And this is, with great power, there must come great responsibility. So my final reminder is that when you're engaging with these data sets, don't forget to pause and seek training to learn about the data before analyzing, to draw on existing resources for reproducible and open science, educate yourself on how to prevent stigmatizing research, and collaborate broadly to ensure that your results are fully contextualized. And with that, I'll close and thank you all for your attention.
Thank you so much, Angie. That's great. Each time I hear these types of items just put together like this, I learn so much and remember so much. So again, it's very appreciative of sharing that with us. As the moderator, I get the opportunity for the first little question, and I guess the one I'll start with is it's the shared data resources are great and all sorts of things can be done by so many people on those resources. Is there any concerns or ways to think about sort of the multiple comparisons, of problem, when 1,000 people go and do similar, even just similar things on, these common resources? I don't know if you have anything to say about how we as a community should be thinking about multiple comparisons or multiple reuses, and do we have a penalty or protection, to worry about in that domain?
I mean, that's a really great question, and one that I'm kind of struggling with myself right now. I'm currently writing a paper where I'm trying to incorporate analytic flexibility, and in order to do that, I've decided that we're going to simply be identifying a subset of pipelines that we feel capture important branching points for our particular study. So we're only testing, I think, 6 different pipelines in that particular study. Multiple comparisons we're handling with just standard corrections at this point, because again, it's only 6 pipelines, and it's something that we can deal with in the standard either Bonferroni or other approaches. But there are other folks that you know of that are running analyses of thousands and thousands of different pipelines, and I don't know what the solution is at that scale of multiple comparisons.
And do
I
you have
think
to pay a
that's,
penalty for their thousands of pipelines?
yeah, like, I mean, and should you have to pay a penalty? Isn't the point there that, yeah, you're doing it thousands of times, and so aren't you really just looking for the large consensus and direction in terms of what the maps then are saying? So, like, I don't know. I'm delighted to know that there are several groups working on multiversal pipelines and developing software to make these accessible for us. I've been in on some conversations to kind of hear where folks are in the process, but I think there are multiple issues embedded in all of that folks are still struggling with, but it's good. We're working on it, right? It's messy, but that's
Yep.
okay.
Yep. And eventually we may have to go back and test some of the hypotheses on new data,
Yeah,
despite the largeness of our existing data does not necessarily preclude the need for eventual new data. So anyway, great.
absolutely.
Other folks out there have questions? Raise a hand or jump in. While I wait, I do have one more queued up. It's a little bit more of a detailed. You presented Ruby Kong's comparison of different anatomic parcellations and things like that. And one of the challenges, most of the questions I ask are because the problems I work on too, or worry about too. Often the anatomic nomenclature that comes from the literature are often not in the atlases that we have. So if someone says dorsolateral prefrontal cortex and FreeSurfer gives you middle frontal, superior frontal, blah blah blah, you still have to end up making up what juxtaparietal lobule is, or temporal parietal junction, or heaven forbid, the default mode network, as an atomic construct. So other than acknowledging, yeah, it's a problem, do you have any insights or thoughts about how that might resolve, or it's just better description of what we do?
So it's a big hot mess. We've seen in other papers talking about the issues related to nomenclature. One of the ones that's the most solutions-based approach is the paper written by Lucina Udine and Thomas Yao and Nathan Sprang. And they put forward a list of anatomically based labels to describe the more common functional networks that we see in studies. And yeah, you're not going to get people to stop calling things a default mode network when it's the default mode network. So, a strict anatomical nomenclature system, I don't think will have a lot of traction. I think in papers, we should refer to the networks that we are describing in terms of both their functional terminology as well as their anatomical terminology to the best of our capabilities. And then I think getting into what is actually the meatier question, which is how are we supposed to do that? That's really hard when we have a lot of atlases out there that have competing network names themselves, when there's sometimes not a whole lot of overlap between those networks if you overlay them on the same map. I think tools like template flow can be adapted to allow us to compare this network in this atlas versus that network in that atlas, and hopefully to overlay like my map or your map into that sort of system so that we can say, well, this looks like 75% of the Dan and the Yo template and 65% of the FPN in this other template. I want to see more accessible tools that allow people to label networks ASAP. I think that is something that's really important because, as you see in the literature, we don't really have a good grasp on what we're calling the networks.
And I think that stifles. Yep, I was going to give it to, and I think that nomenclature is causing part of the reproducibility problems. So yeah, so Felix, welcome to the questioning.
Hi, thanks, Angie. Really nice overview and really important, I think, view that everyone should grapple with. Sorry, my question is, what would you consider in thinking about so longer and structured the most common, let's say, problems that people, let's say, have, or that the errors that people make, and on the other hand, the most severe? And I'm not talking about obvious statistical errors, but the things that creep in, basically. Yeah, I like this result more that. So what's your feeling? What we should look out for ourselves, but as well in papers?
So say a little bit more. What kind of errors?
Yes, kind of problems that are more common in papers and in studies that are done without thinking about the data and how to analyze it. Like just to follow up what you pointed out, but what is basically there's a lot of things that you pointed out, but what are the most common things you have a feeling?
So I didn't really disclose in the talk. I'm one of the site PIs for the ABCD study down here in Miami. So I spend a lot of time working with ABCD data, and that's what pops out in my mind is a lot of folks want to jump into ABCD, but the protocols are so comprehensive and lengthy that a lot of folks aren't really aware of all of the different measures that we have available. So they'll propose an analysis and they'll use this one measure and not really realize that there are several other complementary measures or additional variables that could further contextualize their research question, and they just kind of skip that. Or they do the opposite and they kind of throw everything at it, and it gets really sort of noisy and messy, and it's not as targeted or refined a question. So with ABCD in particular, I think it can be really challenging for new folks to come into that data set and really get a sense of all of the different behavioral and other neurocognition, culture and environment, biospecimens, all the different data that are available to them.
Yes, I fully agree. And I think like Open Neuro, the work what was done, which is amazing because the access of the data we have is still, you got to know the data sets to actually use them in a good way. So it's not only, even though you use data-driven methods, it's very important to be sure. Okay, yes. Sorry, give me a minute.
No worries, don't worry.
Clearly, reviewer 2 is expressing their opinion to his last paper there.
sorry for that, but maybe
Yeah.
one other question. So what do you think is more prevalent, the general, so replication crisis, so because you have both in there, so replication of our results or the overgeneralization? Because I think there's two things that are frequently discussed, and I mean both are in there, but what do you think is the, let's say, the worst we face right now?
I think the overgeneralization, because I think with replication, I think with time and energy, we can really start to pick that apart. But I think with overgeneralization, if we're not collecting data that can generalize appropriately, then we're never going to solve that problem, right? So for overgeneralization, I very much do worry about the limited variability, the limited demography of our samples. That if we continue to just publish studies on white college kids, then we're not really going to be moving the needle on addressing overgeneralizability.
Yes, fully agree. And then I take the opportunity, not too loud here. What would you recommend as being back in the ABCD study? Should there be, I really value the easy way to get the data, but should it be, given the problems, be more difficult to get the data, or should the, let's say, research questions should be vetted more by let's say, experts? Because it's a difficult thing, let's say, because on either side we have the freedom to access it. On the other hand, this problem you pointed out very nicely.
So I don't think that the NIH should be gatekeepers of asking research questions, right? But and actually, you're the first person who's ever said ABCD data are easy to get. You know, it's a little challenging to navigate that portal, right? I think that it is important for every researcher to pay attention when they get access to data to really review their data use agreement and look at those terms and conditions, because a lot of times in all of that boring text, there's a lot of important information that relates to how you use the data, how you manage it. And I know that the NIH is interested in implementing additional training for using ABCD data, just like the Precision Medicine Initiative does, the All of Us study does. So I think that that's a reasonable step, that there should be some hurdles that we have to jump in order to get training on appropriate access and utilization of data. But I don't want it to see it turn into a gatekeeping endeavor where we really have to have our research questions reviewed. I do know that there have been certain studies that have been published on ABCD data that have taken questionable approaches to the research question that really has sort of veered into that shady territory of stigmatizing research. And in those cases, NIH officials have reached out directly to the author to engage in a discussion and to really kind of open a conversation on that topic. I think that's a nice approach, that, you know, they're not gatekeeping, but they are following up and trying to engage directly with the community when they see something that maybe is not a little bit of a disservice to the data. But it is complicated. You definitely don't want the government leaning too heavily into how you're asking your research questions. But I do think there is a responsibility that everybody needs to remember, and that's to the participants' families, particularly in ABCD. These are kids. When they are enrolled 9 to 10 years old, they're giving us a whole lot of their time and their lives, their data, you know, all of the things that to them is personal and sensitive. So we have an obligation, all of us, even if you're not part of the data collection, not only to respect their identity and protect their privacy, but also protect how their data are being used and what we are saying about them broadly.
I fully agree. Thank you.
And presumably some of the reviewers and journals and things like that are also a place where some of this gatekeeping functionality should be imposed, I presume.
That would be nice.
Yeah, but there's always someone who will publish it. Anyway, okay, other questions? Seeing none, I'd like to thank you again, Angie, for sharing this with us. Just so much to keep in mind, and it's great to have it in this nice little presentation.